Hey guys, this is Taxonomy Part 2 from nomorebio.com. One thing I forgot to mention last time concerning prokaryotes are cyanobacteria. Cyanobacteria are also known as blue-green algae, and those are prokaryotes. So sometimes you'll get questions about like which of the following are prokaryotic. Just know that cyanobacteria are prokaryotic or blue-green algae. So today we're going to cover Archaea, and we're going to start to cover eukaryotes. We'll talk about protists and fungi. And then in the next episode, we'll talk about animals, and probably in the fourth part of taxonomy, we'll talk about plants. So Archaea can be a bit confusing. Historically, they've been really difficult to classify, and their classification has gone back and forth between prokaryotic and eukaryotic, now to the point where they're their own domain. So the definition you're going to find on DAT Bootcamp is something like this. I mean, this is literally what it is. Prokaryotic cells that more closely resemble eukaryotes. So it sounds like a contradiction. It can be confusing, but if you remember that, you shouldn't miss any questions about Archaea on the DAT. Because if it asks you, Archaea are considered what type of cells? They're prokaryotic. Why are they prokaryotic? They're prokaryotic because they have no membrane-bound organelles or nucleus, just like a prokaryotic cell. But they more closely resemble eukaryotes for a couple different reasons, one of which is they have histones. So if you're asked which do they more closely resemble, eukaryotes, but what type of cells are they classified as? Prokaryotes, and this is why, because they have no membrane-bound organelles or nucleus. Another interesting thing about Archaea, well, they're single-celled organisms, so keep in mind, I mean, I just kind of imagine Archaea as really similar to bacteria. So they're single-celled organisms. One unique thing about Archaea is they have ether linkages in their fatty acids, so rather than having an ester linkage like we talked about with the prokaryotes, they're going to have ether linkages in their fatty acids, so they would have a glycerol, and they don't have a carbonyl group right here. They just have this ether linkage. The reason that's important is because it enables them to live in more extreme environments, because ethers are less reactive than esters. So the terms halophiles and thermophiles are associated with Archaea. Halophiles are lovers of salts, so they live in very salty environments, whereas thermophiles are lovers of temperature, so they live in very extreme temperatures. So Archaea are typically found, I mean, you might get a question like this on the DAT, where are you most likely to find Archaea? Like deep sea vents is a common place where we find Archaea because it's very hot, so really extreme temperatures, and again, they can live there because of their ether linkages. And then lastly, we don't know at this point, we don't know of any Archaea that cause infection. So while I like to think of them as bacteria, there are a couple important distinctions between them and bacteria. So we don't know of any that cause infection at this point. Moving on to the eukaryotes, so this is you and I. When you think of different organisms, most of the ones you think of are probably eukaryotic. So eukaryotes have a membrane-bound nucleus. That's one really big difference between them and Archaea and prokaryotes. Eukaryotes, if they have flagella or like tentacles they use for moving, if eukaryotic cells have flagella, those are made up of the protein tubulin rather than the protein flagellin, as in prokaryotes. Eukaryotes also have linear chromosomes, so they're going to have histones just like the Archaea do. Linear chromosomes that can condense during cell division. You should also probably know this, that the eukaryotic ribosome is made up of a 60S large subunit, a 40S small subunit, and together they make one 80S ribosome. So that's different from the prokaryotic ribosome. The archaic ribosome is similar to a prokaryotic ribosome, but it's different. So like similar weight, but not exactly. So, and you won't really be asked that on the DAT. So protists are types of eukaryotes. They're the most ancient eukaryotic cells. They are single-celled organisms with plant and animal-like features. Oh, that should say single-celled, not single-celled. single-celled organisms with plant and animal-like features. So if you know that definition, you can get almost every question right about protists for the DAT. A couple other things about protists. You should know that green algae are protists, and amoebas are also protists. Those are like the two typical organisms. You're going to find that basically for every group that we discuss in preparing for the DAT, you're going to have like one or two specific organisms you need to know that belong to those groups. So green algae and amoeba are protists, whereas remember, blue-green algae are bacteria. Green algae are protists. Protists often move using pseudopodia, or podia meaning feet and pseudo meaning like. So they have little structures. They can extend their bodies and move. You've probably seen this before, amoebas moving around using pseudopodia. One other kind of obscure concept or term is an amoebocyte. Amoebocytes are motile. Zoom in a little. So amoebocytes are motile, meaning they can move, and they are totipotent, which we'll talk more about in the future, but it basically means they can become any type of cell in the body. So amoebocytes are motile, totipotent cells found in invertebrates. So if you're asked about what an amoebocyte is, just remember that it's motile. That's probably what they'll ask about, not really so much the totipotent part, but that it's motile, or it can move using those pseudopodia we talked about. Last group for this video are fungi. You can see there's a lot of information here. The DAT really loves to ask about fungi, so we're going to talk about them for a little bit longer. Fungi, typically you're going to think of either mushrooms or yeast when you think of fungi. Those are two really popular examples for the DAT. So a single-celled fungi, an example of that is yeast, multicellular mushrooms. So yeah, you could get questions about which of the following is not true of fungi, and one of the answers would be all fungi are multicellular. That would not be true because some are single-cellular. They have branching filaments known as hyphae or hypha. A group of hyphae are known as mycelia or mycelium. So I'm going to draw a picture for all of this and just kind of show you how I think of it. It may not be the most accurate way to think of it, but it will get you the questions right on the DAT, which most people don't really care about knowing a ton about fungi. They just want to know what they need to know for the DAT. So I'll draw a little picture that will illustrate some of this. Most fungi are saprophytic. So saprophytic means that you feed on dead or decaying matter. That's saprophytic. Some fungi, however, are not saprophytic. They feed on living matter, which the most common example of that is athlete's foot. So you should know both of the things listed right there in that point. Their life cycle follows an alternation of generations. We'll talk more about that in the future when we talk about plants, and I'll remind you at that point that fungi also follow an alternation of generations. So fungi reproduction is pretty diverse and difficult to understand, but we're going to simplify it here. So they can reproduce asexually by budding, whoa, that was really bad handwriting, but it's supposed to say budding, or by something called fragmentation, which I'm going to diagram below in just a second. So budding is how yeast reproduce. Basically a smaller yeast cell just grows off an existing yeast cell. It's different from binary fission, which is, remember, how prokaryotes reproduce. Prokaryotes reproduce by binary fission, where each daughter cell is exactly identical to the parent cell, whereas budding, the cell that came off the parent cell, the daughter cell, has less cytoplasm than the parent cell. So that's how those two things are different. But fungi can reproduce asexually by budding or fragmentation, and they reproduce sexually through sporation, or the production of spores, which are like little sex cells that float around until they find the other sex cell. And we'll talk more about that in alternation of generations again in the plants video. Let's see. Fungi are classified into 3 groups based on the type of sexual spores they produce. If they do not sexually reproduce, they're classified as imperfect fungi. So this is an important point to know. If you're ever asked, like, which of the following is not a classification of fungi, remember that imperfect is a classification of fungi. And the other 3 classifications, I wouldn't recommend memorizing them. However, they're all going to have a word similar to mycelium in them. So if you can remember mycelium is associated with fungi, then you should be able to recognize the other sexual classifications of fungi. And then lastly, this last point, fragmentation, cenocytes, and septate fungi. So I'm just going to draw, this is where I'll draw a little diagram. And again, this may not be 100% accurate, but it'll get the question. It will get you the questions right on the DAT. So we've got some dirt here, and we're going to have a mushroom growing up out of the dirt, and we have to give it spots because mushrooms have spots. So you've got your mushroom here. So the way that you can imagine hyphae or hypha are these filamentous outgrowths, and a bundle of them or a group of them are called mycelium. So fragmentation, like we've talked about fragmentation twice here. Fragmentation occurs when one of these mycelium or hyphae break off and grow into their own new organism. So let's say that this one broke off right here, and it eventually grew into its own mushroom. That would be fragmentation. That's asexual reproduction because there's no gamete involved. It just broke off and formed itself into a new organism. So that's fragmentation. Now, cenocytes and septate fungi are a little bit more complicated. Cenocytes, a cenocyte is a term that's associated with a couple different organisms, not just with fungi. But a cenocyte is a multinucleated cell that results when there's no cell division. So let's say that this hyphae here, this mycelia, grew out and eventually formed into a new mushroom. The mushrooms are getting worse and worse. Eventually formed into a new mushroom. Well, if this, we're going to zoom in right here. If this filament right here is not divided, if there's no wall here, then these 2 mushrooms we're talking about are really one organism. They're one cell because there's no division. So that's a cenocyte. A cenocyte is a multinucleated cell. But if this filament grew out and became another plant and grew a division here, we call it septate. That would be a septate fungi. So these are now no longer one continuous cell with a bunch of different nuclei. These are 2 different organisms, 2 different cells. So that's what's called a septate fungi. So just know the difference between cenocytes and septate fungi, as well as what fragmentation is, how fungi can reproduce asexually. Anyway, that's all the time we have for this video. Next video we'll talk about the kingdom Animalia and cover most of the questions you'll come across in the DAT on taxonomy.